Dear students, today we will talk about amino acids. Amino acids are the micromolecules that act as monomer for protein synthesis. They composed of carbon, nitrogen, oxygen, hydrogen, and sulfur in certain cases. And if you look at the structure of amino acid, essentially they contain two functional groups. One of them is carboxylic group, which impart acidic character. Another one is amine group, or we say amino group, which impart basic character. When amino acid form aqueous solution, that is when you add water to amino acid, the carboxylic group releases H plus ion in the solution, which are accepted by NH2. Since the amino acid contain both acidic and basic group, they are known as amphoteric. They are also known as dipolar ions or sweeter ion. To form amino acid, the four different valencies of carbon are to be satisfied with four different groups. So if you look at the structure of carbon over here, this is the carbon, and you can say one of the valency is satisfied by COH, another one NH2, one by H, and another one is R. Now R group varies in different amino acid. Rest three will remain same and they are present in all the amino acid, whereas R group will be different. Now if you look at the NH2 group, if this is situated at right hand side, we say it is D amino acid, and if it is situated left hand side, we say it is L amino acid. So this represent the general structure of amino acid. Now we'll be talking about the structures of various amino acid. There are 20 fundamental amino acid that exist in our body. Then you have certain derived amino acid, and some non-protein amino acids are there. But we'll be talking over here about the 20 fundamental amino acid which forms the basic structure of protein. So if you look at here, see, the R group is H. This is glycine. R group is CH3 alanine. R group is CH2OH serine. Then R group is CH3OH. Okay, so threonine. And then you can see similarly the R group varies: CH2, CH2S, CH3, then CH2SH cysteine, and then aspartic acid CH2COOH, CH2CH2COOH. So if you look at the structure of, this is asparagine, this is glutamine. Now if you just compare them, so there are amino acids, okay, which are with OH group, such as serine, you can say, threonine. Okay, so these two amino acids are with OH group. They contain OH group, and these OH group, whenever OH group is present, that means it impart polarity, but this is polar uncharged. This is also polar uncharged. No net charge is there on amino acid because charges are balanced over here. Now if you look at the methionine and cysteine, okay, they are the sulfur containing amino acid. Here you can see S group, here you can see S group. So they are also polar and they are also uncharged. Now if you look at the valine, simple R group is there, which is aliphatic. It is non-polar. Here leucine, non-polar R group, isoleucine, non-polar R group. And if you look at aspartic acid, see here you can see two COH are there. One is over here and another one is here. So net will be, total net negative charge will be there on this amino acid because of COOH group, and this amino acid is called acidic amino acid, because if one COOH and one NH2 is there, that means acid and base, they neutralize each other. But if they have additional COH, then it impart acidic character. Similarly, if you see glutamic acid, this is also acidic amino acid. If you see arginine, see here two CO and COOH and two of the NH2. So this is neutralized. Okay. So this is asparagine and glutamine. Same over here. 2 NH2 and 2 COOH. They are neither acidic nor basic. Now if you look at lysine, okay, so if you look at the lysine, you will find two NH2 group are there, one in the R group and another one is the basic structure, sorry, fundamental structure. Okay, so two NH2 group are there, one additional NH2 group is there. So we will say lysine is basic amino acid. Similarly, arginine also contain uh, more than one NH2 group as compared to COOH. So this is also basic amino acid. If you look at histidine, this also contain more NH2 group as compared to COOH. So this histidine is also a basic amino acid. Now if you look at, these are some complex: proline, okay, phenylalanine, tyrosine and tryptophan. If you see here, the benzene ring is present. Now whenever there is benzene ring, they are actually impart the aromatic, they come under the aromatic amino acids because they are all from the aromatic compounds. So phenylalanine, since it has benzene rings, tyrosine, it is also, you can say, having aromatic group, and tryptophan. All these three amino acids are aromatic amino acids. Okay, so this is how you should know the basic thing what is there in their structure once we are studying about amino acid. This is important because on the basis of amino acid, the property of the proteins are decided. Now, if you look at the classification on the basis of functional group, they are divided into basic amino acid, acidic amino acid, and neutral amino acid. So, basic, they have additional amino group. So, lysine and arginine, glutamic acid, aspartic acid, they are acidic. Neutral, they have equal number of COH and NH2 groups. So, rest of the amino acid are neutral. Then on the basis of R group, they are divided into two categories: polar and non-polar. Polar may be charged or uncharged. Charge may be negative or positive. If it is negative, if it is negative, then it is acidic. Okay, and here you have to make some correction, that is this and this. And if it is positive, then it is basic. Okay, uncharged: serine, threonine, asparagine, methionine, glutamine and cysteine. Then non-polar, they are hydrophobic. R group divided into two categories: aliphatic and aromatic. Aromatic are tyrosine, phenylalanine and tryptophan, and rest are aliphatic. So this is how we classify the amino acid. Then there is one more classification which is not based on the structure, but it is based on the requirement depending on the source. Okay, they are known as essential and non-essential. Essential means they are essentially supplied in our diet. It is because our body is unable to synthesize these amino acids. So these amino acids are synthesized by plant, and then through the diet it is supplied it to the human body. So non-essential amino acids are those which are synthesized in the which are synthesized in the human body. Now we will see the structure of peptide linkage. The two amino acids are united by peptide linkage. So here in general, I'll take R1, R2, two different amino acids. Okay. And usually in the body, L amino acids are there. They exist in the living organism. So now the COOH can be written like this, whereas NH2 can be written like this. So OH from COH and H from NH2 is removed, and then linkage is created. So CONH. Okay, this linkage between two amino acid, okay, is called peptide linkage. So this is how, when there is two amino acid, one peptide form. When there are three amino acid, two peptides are formed. Now, what are the functions of amino acid? They help in the protein formation, hormone formation. As we have seen, thyroxine is the hormone where tyrosine is responsible for the synthesis of the hormone. Similarly, tryptophan, which is the aromatic amino acid, help in the synthesis of plant hormone indole acetic acid. Then they act as an intermediate in urea cycle. They also help in the formation of glucose. They help in the formation of histamine, which is a vasodilator. They also act as a buffer since they can resist the change in pH because they have both acidic and basic group. So this is all about amino acid. Thank you.